try. I help me wrong.
Okay, we might get going. Greetings, everyone. Welcome to today's Monash Partners Comprehensive Cancer Consortium Precision Oncology Seminar. My name is Mark Shackleton. I'm the clinical lead of the MPCCC Precision Oncology Program and also the Director of Oncology at Alfred Health. I'd like to begin by acknowledging the people of the Kulin Nation, who are traditional owners of the land on which we gather today at the MPCCC and across Victoria. And I respectfully acknowledge their elders, past, present, and emerging, and I pay my respects to any members of the Aboriginal and Torres Strait Islander community gathered among us this morning. The MPCCC Precision Oncology Program undertakes to build capacity for next-generation sequencing in southeastern Victoria and to build capacities for the application of personalised cancer medicine within MPCCC affiliated health services. In doing so, our program is providing potential treatment options to patients with rare and advanced stage cancers, usually on their last line of therapy. These seminars are a key educational component of our program, and we're really delighted today to be joined by Professor Tony Purcell to share his insights into the tumour immunopeptidome and its role in personalising immunotherapy. So Tony is one of our rock stars at Monash University. He's an NHMRC Principal Research Fellow and Deputy Head of the Department of Biochemistry at Monash University. He's also the Vice President of the Australasian Proteomics Society and a Council Member of the Human Proteome Organisation. His lab works is interested in how the diverse array of peptides presented to the immune system, which have collectively coined the immunopeptidome, is influenced by infection, inflammation, and also by environmental factors. And he's made important contributions to understanding the role of antigen presentation, including characterizing T-cell epitopes in several autoimmune diseases, drugs, hypersensitivity, cancer, as well as infectious diseases. He's actually one of Australia's most cited Australian proteomics researchers and unquestionably a global leader in the field of immunopeptidomics. So it's really fantastic to have you on board, Tony. Now, just before I hand over to him, just a couple of housekeeping things. So could everyone please turn their video off? And even more importantly, please turn your microphones to mute right now. We'll take questions at the end of Tony's presentation. And that can be via either questions that you can post on the chat during his talk, or raise your hand using the typical Zoom function so that I can see who wants to ask the questions. And then after Tony's answered, just lower your hand, etc., etc. Okay, that's it. So without any further ado, Tony, I'll hand over to you now. So maybe if you stop sharing the screen and we'll flick over to Tony. Great.
Thanks, Mark. I don't think I've been called a rock star before, so thanks for that introduction. Let me see. Let's try this one. hopefully you can see my screen. So today what I'd like to do is, I guess, talk about our work in studying tumour immunopeptidomes, and I'll obviously define that for you in a moment for those of you that aren't familiar with the terminology. And really, I guess, take the opportunity to speak about some of our more clinical ambitions with the sort of technology we're developing in my lab in the biochemistry department and the BDI. So what do we mean by the immunopeptidome? So this is a sort of collective term for all the peptide antigens that are displayed on the cell surface that can form targets for T lymphocytes. And what I'm showing you here is a cartoon of the sort of pathway of how these peptides are generated and then presented on the cell surface. So today we're sort of going to almost exclusively focus on this aspect of the pathway, the so-called class 1, MHC class 1 pathway. And this is a pathway where endogenous antigen is degraded primarily through the activity of this large complex proteolytic machinery called the proteasome, which forms peptide precursors that can then be transported into the endoplasmic reticulum, where these MHC or HLA class 1 molecules reside in a peptide receptive conformation, ready to acquire a specific type of peptide. Once that peptide has been acquired, these complexes then egress to the cell surface where they're displayed to CD8 positive or killer T cells. So if you think about this in the context of cancer, if there are changes in protein expression within the cancer cell, and most notably that could be the presence and accumulation of somatic mutations that lead to the formation of neoantigens, and this will be a topic that we'll focus on today. There's the potential to present so-called neoepitopes, so peptides derived from those neoantigens on the cell surface, and alert the immune system to the problem. And, you know, I guess in an ideal world, there would be a T cell that could come along, recognize the cell as being malignant, and kill that cell. And so this is the pathway we study, and we study it particularly by looking at the source of peptides that are on the cell surface. So if you like, the immunopeptidome really is a snapshot of what's happening within the cell. So I've just summarized those points here. And I guess specifically for the talk today, what I'd like to focus on is how we study the immunopeptidome. And I'm not going to go into much technical detail at all, but always happy to discuss that with people if they're silly enough to ask me. To discuss how the cellular microenvironment might influence the immunopeptidome, and to consider novel sources of antigens, which is something that we're particularly interested in my group. And in particular today, I'll explore this class of antigens we call spliced peptides. So just a few sort of quick background points. I'm sure this is known to many of you. The first point is that we are almost certainly will have different HLA molecules expressed on our cells. So we know that this locus in the major histocompatibility complex that encodes these HLA molecules is by far the most polymorphic region of the human genome. We can all have up to 6 different alleles for our MHC class 1 molecules and another 6 for our MHC class 2. And we know that each different allele or allomorph that's encoded has the potential to select a different peptidome. And the reason for this is shown in this cartoon here, where we've mapped for some HLA allotypes, some HLA-A allotypes, where mutations occur or where substitutions occur. And what I hope you can see is that most of them sort of decorate this antigen binding cleft. And this is where the peptide antigen sits in these molecules. And based on the nature of these substitutions, different types of peptides can sit. And so, for example, for HLA-A01 allotypes, they tend to like peptides that have a threonine at position 2, for example, and a tyrosine at position 9. HLA-201, for example, likes a different flavor of peptides, and these peptides often will have a leucine or methionine at their second position of the peptide, and a valine or leucine residue at their C termini. And you'll see some information about these sort of so-called consensus binding motifs a little later in the talk. So how do we study the immunopeptidome? And this is about the level of detail I go into in terms of the mass spectrometry that we use. But we use a process where we immunoprecipitate the HLA class 1 molecules expressed on the surface of the tumour, let's say. We do this under near native conditions. We solubilize these complexes very gently, and we use monoclonal antibodies to capture the peptide HLA complex. We then acid dissociate these molecules, and this removes them from this bead we use to immunoprecipitate with. And it also facilitates dissociation of the peptide antigen from the various components of the HLA molecule. We separate the peptides, and we do a lot of work in this area, and I'll discuss this a little later on. And then we use mass spectrometry essentially just to sequence as many peptides as we can. And you'll sort of see the numbers that we can get to in a few slides' time. The mass spectrometer, in the sort of mode we normally operate in, essentially tries to sequence as many of the most abundant ions that enter the instrument at any given time. There's usually a liquid chromatography step occurring, so with time we see different peptides come through. And then we use various software algorithms to determine the sequences. And this has all been described quite recently in 2019, that's missing here, in a Nature Protocols article. So if you think about the sort of typical strategies for cancer epitope discovery, it can be sort of, I guess, described by this cartoon here, where we take patient-derived cell lines, tumor tissue, etc. And typically one will want to know something about the genomics of that tumor tissue. So that might be by whole genome sequencing, RNA-seq, etc. And in addition, we like to do our immunopeptidomics workflow that I've just described to you using mass spectrometry. We sort of synthesize this information. We get information about genome, transcriptome, and the predicted tumor proteome from here. And we get information about the source of peptides that have been presented. We may use some sort of prediction of neoepitopes based on the genome information to then interrogate our mass spectrometry data and ultimately come up with vaccine candidates. And this is the sort of process that is being used, I guess, typically today by many of the companies like Neon Therapeutics, etc. However, one of the issues that we have with this particular process is that it really fails to predict or fails to account for many post-translationally modified epitopes, such as phosphopeptides, for example, and it doesn't account for spliced peptides either. And this is going to be something we'll discuss a few times. And this is the class of antigens that we've become particularly interested in. So our strategy is a little different. We take our tumor tissue or cell lines. We often treat with different cytokines to expand the sort of the definition of the peptidome, and you'll see why we do that in a moment. We combine the genomics with our immunopeptidomics workflow, and rather than using a sort of template of the predicted proteome, we rely both on that reference proteome, if you like, and also we leverage de novo sequencing, and I'll come to how we do that in a few slides' time. The approach that we take is this immunoaffinity purification approach. We've recently tried to automate this approach using robotics to provide additional offline fractionation. And we're able to, using a cocktail of different antibodies, individually capture class 1 molecules, potentially different allomorphs of class 1. So for example, we have a HLA-A2 specific monoclonal antibody, and the 3 classes of the class 2 molecules as well. So we have a whole series of these tiny little columns, all aligned in series, and we're able to sort of, I guess, deconvolve this sort of complex mixture on the cell surface here, and capture as much of the peptidome as we can. Not all tumours obviously express HLA class 2, but many do, particularly after interferon gamma treatment, for example. And we end up with quite a large number of samples from a single cell line or tumour tissue sample, hence the robotics. So from this, and to sort of give you a feel for where we're at the moment, and from this sort of comprehensive approach, we were able to get quite a large number of unique peptide ligands from each cell line or tissue or biopsy. Obviously, the cell lines afford us a little greater depth. And from this, we're able to do a whole lot of, I guess, mass spec-based tricks, which I won't speak about today. But what it does do is it's really sort of class agnostic, if you like, for the different types of tumor antigens we might be interested in. So we can see the cancer testis antigens, for example, these sort of developmental type antigens that was certainly flavor of the year a few years ago. The sort of much sought after neoepitopes. So these are epitopes that arise from the various somatic mutations that may occur within the tumor, and post-translational modifications. And this includes the spliced peptides, which I'll describe in more detail shortly. So why might we want to look at different cellular microenvironments? And this is just a snippet from a recent paper, and I would sort of direct you there to read more about this type of analysis. But the reason that we're interested in looking at different treatments is that we see quite a lot of plasticity in the sorts of peptides that are presented. And so we can see here is that in the presence of interferon gamma, we have almost 50% of the peptides are unique to this one condition. So by combining both sets of conditions, we obviously get much greater coverage of the potential immunopeptidome. And I think all of these different types of treatments are obviously relevant to different types of tumor microenvironments as well, and the sorts of antigens that might be presented. We became interested in spliced peptide antigens as a source of tumor-specific targets. And really this was something that's been known since, I guess, around 2004. And we'll discuss the details of that a little later. But it wasn't really until this paper in 2016 from Michele Misto and his colleagues that speculated that a large fraction of the immunopeptidome, a third of the peptides on the cell surface, originate from this so-called splicing. And there was this, and what do I mean by that? So one can consider that, you know, every protein that we understand that's templated by the genome is a linear sequence of amino acids, and if they're degraded normally, one might consider that this sort of long string of amino acids, if you like, is clipped by the proteasome at different areas, generating these various fragments that are then transported to the cell surface by MHC class 1 molecules. However, we also understand that in the proteasome, under certain conditions, a sort of reverse proteolysis can occur, or a transpeptidation reaction, where these two discontinuous segments from this antigen can be ligated together. And so you basically form a totally novel chemical entity of novel sequence. And this can happen in many different ways, and I'm not going to go into a lot of detail today, but this type of splicing is called cis splicing. This can also occur in reverse, so this part of the molecule, this C-terminal fragment could be ligated onto the N-terminus of the fusion peptide, of the spliced peptide. And it can also occur between two different source antigens, so-called trans-splicing, or the formation of a hybrid peptide. We decided to take a different approach, as I mentioned, to identifying this particular class of antigens. And in our approach, we sort of combined the best of both worlds, at least I think so. And that is to leverage de novo sequencing of MS spectra. So basically using the information inherent in the mass spectral data we collect to assign high confidence sequences, and also to use a reference proteome to assign as many of the spectra to sequences based on that reference proteome. So we end up with a large number of library hits. So these are peptides that are templated. We call them linear peptides. And also a proportion of very high quality spectra that do not match anything in the reference proteome. And these are the ones we really interrogate for a potential assignment as a spliced peptide. And that's sort of, I guess, recapitulated here in this workflow where we anticipate each peptide will be linear. If not, we ask, is it a cis-spliced peptide? And if not, is it a trans-spliced peptide? Or is it something else that we just can't assign? And when we use this analysis, we recapitulate the observations made by Mishta and colleagues a few years earlier in Science. This was published in 2018 in Science Immunology, where we see for a number of different alleles, quite a high proportion of peptides that are best described as either linear, cis, or trans-spliced. And that proportion seems to vary based on the type of HLA allele that we look at. So I'm not going to dwell too much on published data. I thought I'd talk about something a bit different. And this is sort of a little bit of back to the future, if you like, and describe some recent work that we've been doing. So what we wanted to do was ask, can we recapitulate previously published immunogenic spliced cancer epitopes? These are ones that have been sporadically reported in the literature since about 2004. By accessing through collaborations the cancer or the cell line derived from that cancer where the spliced epitope was first reported, and ask, can we use our algorithm to actually detect these and really sort of, I guess, validate our approach? What we were able to do is to sort of look at each of these different cancer cell lines and again, I guess, demonstrate this observation that somewhere between about 20 and 30% of the peptides are best described as being spliced in origin. And we were able to identify all of the previously reported spliced peptide epitopes. So this epitope here is from FGF5. It was reported by Kenichi Hanada, John Udell, and James Yang in Nature in 2004. And really the approach they took to find this was quite heroic, where they took basically cDNA libraries and tried to truncate the identified antigen down to the minimal epitope. And at some point they lost the ability to do that. And it was only through some quite imaginative peptide synthesis they were able to identify that this was actually a spliced peptide containing two different regions of FGF5 stitched together by the proteasome. Here's another example from a different cell line and a different study published in 2014 by Benoit Vandeneind and colleagues, where again, there's this Pmel derived fragment which contains a large proportion of the Pmel with a small addition at the C-terminus here of an arginine residue. And again, what I hope you can see here is that we can elute this peptide, we can match it very nicely to the sort of fingerprint, if you like, that we see with the synthetic peptide. And again, our algorithm seems to be very good at identifying these particular spliced peptides. So now to, I guess, some more novel data. And one of the things we see when we look at some of our favorite melanoma-associated antigens, for example, and this is again Pmel, is that we see there is a region of this particular antigen highlighted here in red that seems to be constantly presented with a heterogeneous N or C termini. And these peptides can either be linear templated, so this is the linear sequence, as you can see here from the Pmel antigen. But in addition to those, we see quite a number of cis-spliced peptide variants here, where we see this same core sequence with various N-terminal additions from different parts of the molecule. And again, the only explanation that we can come up with this is that these have been spliced together, presumably in the proteasome, using the mechanism that I've already described. Again, we're able to go through. We've done peptide binding assays to generate real binding data in collaboration with Alex Sade and John Sidney at La Jolla. And we've also been able to match all of these peptides to a synthetic variant to sort of fingerprint, if you like, that our experimentally acquired spectra matches very nicely to the spectra we see from the synthetic peptide. As part of, I guess, a sort of translational effort in the lab, we started talking to Jerry Lynette and Beatrice Carino from the Parker Institute in the US. And their laboratory is very interested in making dendritic cell-based vaccines. And I'm sure I don't really have to describe this to you, but essentially this process is of taking dendritic cells derived from PBMCs of patients, pulsing these with antigen or peptides in our case, and administering them back into the patient and looking to see whether those dendritic cells are able to initiate an immune response to the peptides and potentially also have an impact on the cancer in the particular patient. And what I thought I'd share to you today, this is sort of very recent, very exciting data for us, is the sorts of responses we're getting to some of these spliced peptides. So here we're pulsing with a number of different spliced peptides shown here on the x-axis, along with some control peptides here. So both of these are peptides that either contain mutations or have a modified C terminus to improve the binding affinity of particular regions of Pmel. A control peptide from flu nucleoprotein as a, I guess, a control for immunogenicity. And what I hope you can see is that a number of our spliced peptides here are actually giving very nice cytokine responses from PBMCs from the patients following the dendritic cell vaccine. And that's just sort of compiling all the data together to sort of show you which of these particular peptides seem to be eliciting immune responses. So we don't only see spliced peptides from Pmel. In sort of some of the data we've looked at, we see a number of examples of spliced peptides from additional antigen sources. Some of these are listed here. And it really begs the question, why would the host cell go to such efforts to sort of scramble proteins and display them on the cell surface? And the next two slides maybe give some insight into why this might be. So if we look at one of the patient-derived cell lines that we've studied, it's a melanoma cell line. And what you can see here is a list of a number of melanoma-associated antigens. You may not be able to read them, but really it's just looking at this scope, if you like, of different antigens. What I hope you can see is that in blue are the antigens where there is one or more linear peptides, so templated peptides from each of the antigens. So, for example, if we look here, this is MAGE-A4, and you can see there's a number of linear peptides here, around 20, derived from this particular antigen, and there's a few more that also come from this antigen that are best explained as spliced peptides. So this antigen has pretty good coverage with the linear, and the spliced peptides have maybe an additive coverage to it. But if you look at some of these other antigens here, there are no appropriate linear peptides that match the sort of HLA binding motif of the HLA expressed by this particular cell line or patient. And so what's happening is that splicing is sort of allowing coverage of these antigens. And one could obviously see that that would be very important if you wanted to broaden the sort of repertoire of peptides available for T-cell recognition. So perhaps this is a reason why this sort of process has evolved. And if you look at how interferon changes the repertoire of these, what I hope you can see here is that quite dramatically we can see, for example, an interferon gamma treatment. So here's another potential antigen. This is one of those acute phase antigens where extremely good linear peptide coverage under basal conditions, but as soon as you induce immunoproteasome expression and interferon gamma treat, suddenly you lose all of the linear peptides and you're only really left now with this spliced peptide here to cover this particular antigen. So again, really sort of showing that not only is splicing able to broaden the repertoire of peptides available for T-cell recognition, when that tumour is infiltrated, for example, and there's a pro-inflammatory environment, it's also able to provide coverage of that antigen when the linear peptides disappear from the repertoire. Presumably they're destroyed during antigen processing. So just to sort of quickly summarize what I've just told you, I've discussed briefly how we can identify very large numbers of these peptides now, that we see a high proportion of these peptides are cis-spliced, and if you add the trans-spliced peptides to that, it becomes even higher, 20 to 30%. Many of the spliced peptides, and the ones that we're particularly interested in, are derived from melanoma-associated antigens, and I'll show you data now where we can show these spliced peptides aren't just immunogenic by taking PBMCs from patients and re-stimulating them and driving them hard in culture, as we showed in the cancer immunology research paper, but also we've shown that they're immunogenic following ex vivo expansion after dendritic cell-based vaccination, suggesting that not only are they appropriate targets, but they are also immunogenic in humans. But what I'd like to do in the remaining time I have is sort of talk about, well, it's sort of fine to have fantastic technology, but it's fine to work with patient-derived cell lines, but how are we really going to take this and become more clinically relevant? Because these patient-derived cell lines have been in culture, some of them for many years, and probably their resemblance to the primary tumor they were derived from is not so great. And there have been some efforts recently to sort of look at this, both in terms of using organoids derived from single cancer cells. There's a nice paper from Albert Heck's lab just reported in Nature Communications. Using PDX, so patient-derived xenografts, so tumours grown in nude mice. We've reported that in collaboration with Mark, and others have also reported the use of PDX tissue. And the other question we wanted to ask is, can we move from this next generation sequence-based prediction of neoepitopes to formulate vaccines to being able to at least validate neoepitope presentation on the surface of the biopsy? So I'll show you two sort of snippets of how we're approaching that. One is a peptide barcoding approach that we call peptide PCR. And it literally is a way of amplifying the signal of peptides from very small amounts of tumor biopsy, for example. And the last example I will hopefully have time to show you is I guess, leveraging some very new technology, a new instrument that we have on loan in the laboratory, which has far superior sensitivity and looks like it may be able to boost our ability to work with small amounts of material as well. So one of the big hurdles, of course, is having enough material. So when we work with cell lines, we're typically working in this range here, somewhere around about 10 to the 8 cells or so, which is about equivalent to a 1 gram biopsy. Now, I guess in our experience, one doesn't always get a 1 gram biopsy, and I'd be interested to hear, I guess, from you whether that's a possibility. But typically with biopsies, we're probably working in this range here where the amount of material probably isn't sufficient to give us particularly good signals in the mass spectrometer. So Sri Ramarathinam, a postdoc in my group, was really, I guess, challenged with how do we get around this sample size limitation? And then Sri came up with a very innovative approach to do this using this peptide signal amplification strategy, or peptide PCR, to validate neoepitope presentation on cancer biopsies. Now, this work's been put into bioarchives, and it's currently bouncing around a few of the high impact journals trying to find a home as we speak. So the challenge really is with these low abundance peptides and small amounts of material. Now we use a MS spectra essentially as a fingerprint. So when it's intense, we get a very nice fingerprint and we're able to identify that peptide quite nicely. So akin to the sort of analogy suggested here of a criminal database. For low abundance peptides or from peptides where they're derived from just not quite enough material, we get a partial print. And this often isn't enough information to really confidently identify this particular peptide in this case. So the approach we took was to do our normal small scale, and we've miniaturized this now, affinity purification of the HLA molecules, to isolate the peptides and to barcode them. And I'm not going to go into the details of how this barcoding works today, but essentially what we use is an abundant source of peptides of interest, and we put a particular barcode on those. And then we take eluted peptides here, we put another barcode on them, and then we analyze them in a mass spectrometer in a way such that the signal from the so-called carrier peptidome, or the abundant source of these peptides, is used as like a primer, if you like, to amplify the signal from the endogenous peptides on the tumor. Now, what does that look like? So here now, I'm going to show you data, which is an immunopeptidome of just 1,000 cells. And what this does, this was for a B lymphoblastoid cell line, so a B cell lymphoma, if you like. And we're able to recapitulate the properties of the known HLA peptides from this particular cell line. So in this example, we used a carrier peptidome from the same cell line. So we're able to amplify the signal from 1,000 cells using a more abundant source from 1,000,000 cells. And even at quite high stringency, so a false discovery rate of 1%, we're able to detect around 900 peptides in total. And if we look at this assay and say, well, if we titrate in the amount of material we start off with, you see quite a nice linear relationship here between the input of cell numbers, so from 5 million cells down to 1,000. We can see the canonical length distribution for the class 1 peptides here in brown, so 9 to 11 meters typically. And for the class 2 peptides, also expressed on this cell line, typically 14 to 18 amino acids, so again, very nice. And we can deconvolve the spectra and show the HLA binding motifs for the key HLA molecules expressed by these particular cells. So it all look really nice in a sort of contrived system where we can control everything. What about for a real life biopsy, if you like? And in collaboration with Mark, we were able to access a small biopsy, around 20 milligrams, for which part of that biopsy had been grown up as a PDX in nude mice, as shown here. We had exome sequencing information, so we could make a patient-specific protein database. And we were able to barcode our material here using material from the PDX, which was substantially more abundant as the carrier peptidome to amplify the signals of the peptides contained within the biopsy. And this experiment, we sort of explored the amount of the carrier we could use. So peptides derived from 400 milligrams, 150 milligrams, or 50 milligrams of the PDX. And you'll see on the next slide, this was certainly too much. And then peptides derived from most of the residual biopsy, and then a very small amount, 1 milligram of biopsy, which is where we're hoping to get to in this particular experiment. And again, what I hope you can see here is that this was also quite a successful approach. We were able to detect around 1,400 peptides, 900 from the 1 milligram biopsy sample. So again, akin to that 1,000 cell sample from the BLCL. These are all class 1 peptides because this particular cell line doesn't express class 2. Again, you can see typical length distribution of these peptides, which is encouraging. But even more encouraging was when we started to look at the peptide identities. And what you can see here are peptides that are derived from a number of well-accepted tumor-associated antigens from melanoma, including things like tyrosinase, MAGE proteins, etc. So we think that this technology is going to be very useful for validating predicted epitopes in primary tumor cells from biopsies. And one of the approaches that we're very interested in doing is to, rather than use PDX, which is obviously a fairly time-consuming way to generate a carrier, to use synthetic peptides based on the epitope prediction algorithms, etc. I thought I'd just very quickly show you an example that we've just generated very recently using a peptide library to probe a number of primary dissociated cancer cells. We were working with about 1 million cells total in the samples. And so from a relatively simple peptide library of around about 187 peptides, we're able to find very good evidence for well over 2/3 of those using this same approach. The data here is shown as a heat map, so the brighter the color, the more intense the signaling. You can see, we see peptides from our carrier here that are seen differentially across the different types of cancers here. And also we can see peptides in the cancer that we don't see in the carriers as well. So some particularly abundant ones that are amplified through their presence in all of the different cancer samples. So we think this sort of technology is going to be very useful for asking a series of different questions, and at least in cancer, asking whether particular tumor-associated antigen-derived peptides are present on the cell surface, which could really guide potential therapy. And clearly has applications in vaccine trials, etc. as well. And we're certainly very confident now that we can get down to milligram quantities of biopsy, and I think we're going to be able to start working on things like fine needle aspirate biopsies, or biopsies where there's a very low percentage of tumor cells. Now, in the last couple of minutes, I just thought I'd share some very recent data we've generated on this particular instrument here. Oh, there you go. I forgot about Shri. So this is work that the data was acquired by Kirti Pandey, a PhD student in the lab, on this Bruker timsTOF instrument that we've been loaned from the vendor to sort of put it through its paces. We had a lung cancer biopsy. It was a fairly small biopsy. And we decided, rather than do this sort of fancy manipulation of the sample, what can we do just using a more sensitive instrument? And much to our delight with this particular challenging biopsy sample, again, we were able to get around 1,500 peptide sequences from this particular biopsy sample. And we could deconvolve them and find various class 1 motifs based on the immunogenetics of the patient. And I think this, again, is another example of just whereby using, unfortunately, a much more expensive but very sensitive instrument is going to be able to give us insights into these sort of important clinical samples, and move the work we do away from cell lines to actually looking now at sort of real samples, real life samples, if you like, and clinically extremely relevant samples. So I'd like to just acknowledge the people that I've failed to do throughout the talk. So particularly the individuals from my lab, Puya, Nathan, and Chen really sort of drove the spliced peptide algorithm work. Shri, I've mentioned on a number of occasions. Richie also involved in the spliced peptide work as a master's student, now a PhD student in the lab. Kirti, I've mentioned. Mark, we've worked with to access the biopsies and really sort of think about how we can use this translationally. Ralph is head of the mass spec facility at at BDI, but in a past life, he was a postdoc in my group and actually started to generate some of this data as a postdoc way back then. I've mentioned the peptide binding work with Alex Sete and John at La Jolla Institute for Allergy and Infectious Disease. I showed you some of the really exciting translational collaborative work with Jerry and Beatrix from UPenn. We've worked closely with Andreas Behrens for the melanoma cell line, patient-derived cell lines. And we've worked very closely with Bioinformatics Solutions on their de novo sequencing algorithms. I'd like to thank the funders of the work and thank you for your attention. Hopefully I've left enough time for a few questions.
Great, thanks very much, Tony. That was a fantastic, incredibly exciting technological advance, I think. So we've got time for questions. So the options are to stick your hand up or to put something in the chat function that I'll try to respond to. So Tony, I wonder if I could sort of kick off the discussion. So what's the relationship between immunopeptidome abundance, and I'm talking about those peptides that are actually presented on the cell surface, and immunogenicity?
Yeah, look, that's probably the $64 million question.
But sort of, it sort of relates to the issue about this, about the sensitivity of detection of exactly what's in there, and do you really want to detect every single peptide, or do you only want to detect the more abundant ones? Because it's almost one of the key questions that relates to the clinical translatability.
Yeah, so look, I guess it's how you want to define immunogenicity as well, right? So I think when you're seeing a patient, for example, you might argue that it's a failure of the immune system to eradicate that cancer. So I guess the way I sort of think about it is, can we identify ideally abundant targets on the surface of the tumours that are cancer specific and for which we could vaccinate and generate immune response to? I think most people measure pre-existing immunity, and that's a good proxy, I guess. But I guess there's been a number of clinical trials using cancer testis antigens, for example, where robust immunity has been generated, but it hasn't really touched the tumour. So I think it's generating the right type of immune response to the right type of target. And I think there will be some of these targets that will generate an immune response spontaneously. And there'll be other targets that, for whatever reason, aren't particularly well cross-presented in the draining node, for example, and do not or fail to elicit a strong enough T cell response. And that's the sort of class of antigen that I think perhaps the spliced peptides might fall into. And I think if we can augment a therapy with some sort of vaccine approach to induce potent T cell killing, then I think we can up the chances of actually having an impact on disease, and that's I guess the thinking we have with Jerry Lynette and Beatrix Carino, for example, where I think the next steps in that study are that they now have the TCRs. We're going to put those into PBMCs and ask, do they kill the tumor? Unfortunately, the patients that get these DC-based vaccines are pretty end-stage disease. So we're really just looking at immunogenicity. We're not seeing a big impact on their disease, which is generally metastatic and widely dispersed and whatnot. But I think that's the sort of path I see this taking. I guess to answer your question more directly, there doesn't seem to be a strong relationship between abundance and immunogenicity. If we look at infectious disease, for example, where we can look at how much of a viral epitope is presented, and is that the best epitope or the one that's most abundantly responded to. And there seems to be a very complex relationship there between stability of the complex, abundance on the cell surface, and at what point of infection does it actually come to the surface. You know, and I think there you can look at the direct infection of the target. You also have to consider, well, what gets cross-presented and primes T cells. And then you can look at the ultimate immune phenotype of which, you know, if you take tetramers, for example, and look in PBMCs from convalescent individuals, which ones generate the most memory, which ones have the most abundant, what's just the most abundant specificity out of the CD8 positive T cells. And that seems to be different for each virus and a lot more complex than a simple rule. And you know, I always assume the simple rule might be the most abundant peptide is the best. Not so. And we've worked a lot on flu, for example, to try and dissect that in recent years.
Great, thanks. So any questions? I'm looking for hands up. Not seeing any at the moment. Nothing in the chat. That's
what happens when you give a seminar near December.
On December on a Friday afternoon after COVID.
That's right. Exactly. Perfect storm.
Epitome of the graveyard shift.
Yeah.
Great. All right. Well, unless any questions are going to pop up, we can probably close it off then. So just first to say thanks very much, Tony, for such an excellent talk about a super exciting innovation coming out of your area, and really excited to see where that leads. Certainly, thanks everyone for coming to the seminar today. We've had great attendances generally during the year, and this one's been no exception. There is currently in planning a series of similar seminars for 2021, and more information will be rolling out to our mailing list about those. And just remind you all to sign up to receive the MPCCC newsletter, or to follow the MPCCC on socials, which is really the best way to stay updated about the activities coming out of the program. All good. So
okay,
have a great weekend, everyone.
Well, thanks everyone.
Thanks again, Tony.
And
see you in 2021.
No doubt. And look, if anyone does have a clinical problem, they're interested in this sort of technology, I'd be very happy to talk to people. And clearly, you know, access to patients is something that we find difficult, and a number of you are dealing with every day. So you know, it'd be really great to sort of cross that void and start to think about doing some translational work within MPCCC.
Fantastic.
Fantastic. See you, everyone. Bye-bye.
Great.
Thank you very
Thanks,
much.
Tony.
Bye-bye.
Yes, thank you.